Okay, ready to go. So I want to talk about digestion. So whenever, for example, honeybees consume food, they're actually feeding the microbes. And the microbes are digesting the food and secreting microbial products called short-chain fatty acids. And short-chain fatty acids are the raw fuel for all of the metabolic processes. For example, digestion, reproduction, longevity, immunity, work. Energy is a budget. It all depends on what you spend it on. So this slide shows a really interesting experiment that was done comparing two different sets of bees and their diets. So one diet that the bees were fed was germ-free diet. Essentially, it was an artificial diet with pollen substitute and sugar syrup. And all of those, all that diet was sterilized, so it didn't have microbes. And they compared, and this particular experiment compared bees to that were eating conventional diet, were eating a natural pollen and natural microbes and natural nectar that had beneficial microbes on them. And what they have noticed is that with microbes, you are able to generate much better digestion and much more variety and abundance of short-chain fatty acids. And this is actually shown here that with conventional diet, with beneficial microbes, we are able to generate better and more of these of this raw fuel, where we optimize digestion, we generate as much energy as possible from it. Okay, next slide. There you go. Additionally, we're seeing that bees do way more. The bees that have received beneficial microbes do have more body weight. And we see that through analyzing body weight, we see that a big improvement of it is through, a big weight gain is through vitellogenin. And vitellogenin is a very important glycolipoprotein that is imperative for overwinter survival. Additionally, varroa mites are eating vitellogenin. That's the main bee fat that they're feeding on whenever there's a mite infestation in the hive. So yes, we are able to treat for mites with different mite treatment products. However, if the bees are strong and more robust, they are able to tolerate that challenge a little bit longer and stay healthier until you are able to do mite treatments. So bees that have received probiotics are much stronger and fatter bees. And this is a prime example of it. The bee on the right was treated with probiotics. The bee on the left was not. He received a lot of conventional artificial diets, and some of that diet was not taken up. It was not properly digested. And sometimes if the colony is not very strong, it will not take up the entire pollen patty there, where the pollen patty will crust up and we'll have to throw it away. Same thing for syrup. If the colony is not strong enough, the syrup will not be fully consumed and it may ferment. So once again, more waste will be generated. So I've talked about nutrition. The next topic that I would like to talk about is detoxification. Bees are pollinators, and whenever they go, for example, on either pollination contracts or they will visit orchards that are in your neighborhood, they were potentially sprayed with pesticides or herbicides or fungicides. All of those agrochemicals do a negative effect on the gut microbiota. Or sorry, not on the gut, on the health of the honeybee. So what I would like to show this here is a really interesting experiment that was done that was done to expose the honeybees to a neonicotinoid and a fungicide, thiamethoxam and boscalid. Those are the common agrochemicals that are being used in our industry right now. And so this particular graph shows control bees, And we are seeing that these are quite healthy when the package is installed, for example, and they very slowly start to dwindle as the time progresses. However, with the addition of the fungicide and a neonicotinoid, these crash fast. At day 20, we're less than 60% survival. Eventually, those bees died fast, and bees are not supposed to live 20 days. They're supposed to live much longer than that. However, with the addition of beneficial microbes, we're able to save those bees from the exposure of pesticides to them. And this is huge because it is much easier and cheaper and safer to replace the microbes in the gut and have them take the fall of the pesticides as opposed to the whole colony. Because the common mode of action of pesticides is to turn off the detoxification system of the target organism. Unfortunately, bees have the same detoxification system. And with the beneficial microbes, we're able to keep that detox system functional much longer. Not only do we do that, but we also bind and break down pesticides. Now, there are many different species of probiotic that will do pesticide degradation, that will help with pesticide effects. And since we live in the age of modern agriculture, probiotic properly or survive long. Ooh, my connection is a little bit unstable. Sorry about that. Okay. So another topic that I like to talk about is protection. Protection is quite interesting because bees have an intrinsic defense. They have an endogenous defense. They do have an immune system. However, on top of that, they have a microbiome defense. Think of it as like a microbial shield or a microbial sponge, okay, that is present on top of the innate immune system. And this microbial biomass is used as essentially a first line of defense against bacterial infections, fungal infections, gut parasites, and as well as environmental pollutants. I mean, all of these agrochemicals that we use can be absorbed by microbes as a sponge. Now, with age, with heat, and with different stresses that are present on that the bees are encountering, we're losing this microbial shield fast. As you can see from this slide, not only pesticides, but poor nutrition, varroa, viruses, bacterial infections, nosema, it's death by 1,000 cuts. And bees are just such hardy troopers to tolerate all of that, but not all of them can do that. And most of them will crash because of all of these challenges that we're encountering. Oops. So with beneficial microbes, we're able to repair it. But I would like to show you another example of how, just a visual example, how agrochemicals actually affect the microbiome. So we performed a particular experiment taking honeybee gut sample. So essentially we crushed the honeybee, we took the contents of the gut and we plated it on a petri dish. Essentially we grew those microbes on a petri dish, okay? In the middle of that petri dish, we made a hole and we placed particular antimicrobial substances inside that hole to see how they inhibit or affect the microbiome. So about 48 to 72 hours later, we were measuring essentially the zone of inhibition of how these microbes, how these agrochemicals affect the microbes. So what we have found is the following. For example, when we use essential oils, wintergreen oil that is present, for example, in multiple essential oil supplements, Honeybee Healthy or other essential oil supplements, it's totally benign on the microbes. There's no zone of inhibition. When we tested, a Roundup, which is an herbicide, it did have some antimicrobial activity. Okay, fumagillin, a very commonly used or prescribed nosema treatment, is essentially a broad-spectrum antibiotic. It does reduce the gut microflora. Not only does it do that, it decreases the hunger of the honeybees. It takes about a week for them to recover that hunger. Is one of the antibiotics that is used by beekeeping industry, and as you can see, there's a huge zone of inhibition. So antibiotics or anti-biota, anti-microbes, they will kill beneficial microbes. So what we have is we have a complete microbial protection. We make probiotics for honeybees. And what we are seeing as a result by using probiotics, we're seeing a cleaner hive because we are not only installing a microbial shield, we're using microbes as a sponge to absorb all these either agrochemicals or negative side effects. And we see longer living bees, and we see reduced comb contamination, and we see an economic improvement of about 5% on a continuous use. So I want to briefly talk about our location. We're located in Milwaukee, Wisconsin. We are also urban beekeepers. We have bees on the roof of our manufacturing building. This is our apiary, and where we test their products, and we also raise bees and collect honey. Urban honey is quite good because everybody gardens, and very few people use pesticides. So it's quite a clean environment to raise bees. This is our blending facility where we formulate our products. We have a fermentation facility, and when we grow particular species, we then either freeze dry it or vacuum dry it, depending on the species that we're growing. And then we make blends that are appropriate for a particular species. So this particular one is for honeybees. Our facility is safe food, safe feed certified, and this is really important because everything that we do is traceable. We know exactly where all of our ingredients are coming from and where exactly they're going. And this is really important because just in case there's any problems, we can solve them immediately. Thankfully, out of our 10 years of manufacturing excellence, we had 0 problems, and we intend to keep it this way. This is the team that works with us, and we are really grateful to work with these wonderful individuals. You can see this myself, it's my wife Vera. These are EAS Master Beekeepers Earl and Carol Hoffman. They were instrumental in the creation of this product. Dan Williams is our production manager. Renee, who is also on the call with us today. George is our fermentation plant manager as well. Nick, Ben, and Garrett are production guys. Sandra is also a fermentation specialist. And John is our newest addition, that he is a beekeeper and a field support specialist as well. So everybody on this team, I'm so grateful and proud to work with, because I am unable to do any of this work without the help of them. So thank you, all of you. So going back to Super-DFM Honeybee. Super-DFM Honeybee, we ask you to please feed it dry, because the products that the microbes that we use are lyophilized, they're freeze-dried, and the lactic acid bacteria is anaerobic. It does not like oxygen. We activate or we wake up these microbes by addition of moisture. Now we want them to wake up exactly where they should, in the gut of the honeybee. The gut of the honeybee has moisture on the mucosa, on the mucus layer of the intestine, and it has minimal amount of oxygen. It has 2% oxygen level within there, and this is a perfect environment for our lactic acid bacteria or for microbes to just wake up and hit the ground running. Please use every 30 days, and the reason for it is to inoculate every brood cycle. Everything in nature is dynamic. The gut is dynamic. Lots of things change almost by the minute. So by using every 30 days, you are inoculating every brood cycle with beneficial protection. This particular video shows Carol, and she is applying dry products, dry Super-DFM right on top of the brood chamber. If you have honey supers on, you actually on top of the honey super, it doesn't matter. But in this particular application, we will be using a slightly different. Erica has a really awesome device, which I think is phenomenal, because not only it's able to inoculate with beneficial microbes, but it has multiple other functions that I will allow Erica to describe instead of me. But I think it's a really awesome product, and we're very excited for it to be coming out, because I think lots of beekeepers in the world will be benefiting from it, and we're very excited about also putting it in all of our hives and our apiary. So the most important thing is that this product is very stable. It's able to it does have a 5-year shelf life, and it's very palatable. Bees consume it right away. So without further ado, I like to conclude my presentation and ask as many questions as possible. And answer as many questions as possible. And this picture is a part of our fermentation facility where we grow our microbes. It's the same thing as essentially a brewery, but we don't focus on alcohol. We grow microbes. We focus on bacteria. So and this does conclude my presentation. I am going to stop sharing the screen and yeah, move on. Just a second. Pause.
Great. Slava, thanks to you so much. That was really educational for me to kind of hear about the entire story and everything that's happening. I think we did have John just joined us, and so I don't know if you're able to turn his video and go ahead and just tell our participants. Yay, there we are. Our participants, I'm going to just talk a little bit about what we're doing with Best for Bees, bee vectoring, and why this actually has tied us in with strong microbials, where we can collaborate in this and have their product that we're using in our product, which is kind of neat. To not many joint ventures in the beekeeping world, so it's great to have that moving forward. And in the meantime, while I'm talking, if you've had any questions, or if you have any questions once I'm talking, go ahead and you can put them in the chat. And once I finish up, my portion here is fairly short. We're going to open this up, and like I said, kind of as a chat, so you can put your questions there, and then the Strong Microbial team can also talk to us about some of the things that they've found or have done, or just kind of see a flow of this of how it goes, so that we can just basically hear all about these probiotics. So I am going to share my screen, and we're just gonna hold on, a slideshow. All right, and okay. Are you guys seeing my my screen there? Are you guys seeing that there, Renee?
Yep, I can see it.
Looks good. Okay, perfect. All right, so I'm trying to hit view slideshow, and here we go. There we are. Okay. All right. And is that showing as notes, or is it showing as the slideshow?
Yeah, it's a slideshow.
Okay, perfect. I mess that up sometimes, so I just want to make sure I got that right. All right, so I did introduce myself at the beginning, but I'm going to go ahead and introduce myself again for those of you that came in a little later, or you maybe just weren't paying attention. My name is Erica Shelley. I am the CEO and founder of Best for Bees, which is a honeybee and bumblebee research company. And just like Slava was saying, he's a scientist. I am as well. So I got my PhD in molecular medical genetics. I actually worked in fungi before I moved over to honeybees. That zone of inhibition stuff, definitely have done a lot of that in my time. And it's really fun to see what's going on with that. But what we've been working on over the last 2 years was this really neat project that came up between us and the Peter Kevin Lab at the University of Guelph, dealing with a phenomenon called bee vectoring. And you might not have heard of bee vectoring before because it's brand new, but it has been being used just for the last couple of years for delivering protectants out to crops such as strawberries, blueberries, crops like that to protect them against pests and diseases. And so this, this technology basically has a bee walking through a powder. And then as the bee visits flower to flower to flower, it drops the powder behind just during its normal pollination visits. So the bees are working instead of having, you know, a machine out there spraying these things. You know, the bees are basically doing the work. And the nice thing about that is that the bees are always working day in, day out. You just have to go in and you can use them to deliver these powders to the crops. Now, the powders are usually soil fungi, what we call entomopathogenic fungi, and meaning that they basically go in and they eat up, they don't eat up, they sort of go inside their host, and then that fungi starts growing inside the host. And the host can be a fungus, it can be an insect, it can be whatever. It is able to actually penetrate and start dividing in. And so we realized that we had the ability to perhaps use this bee vectoring to move entomopathogenic fungi into hives in order to deal with pests and diseases that we find inside the hive. And particularly when we started this project, we were looking at varroa mites, because that's the biggest issue that we tend to have here in North America in terms of pests inside of hives. So we developed a product called the ProtectaBEE, which directs traffic into one side and out the other side. So these cones, basically they're red, which makes it a little bit hard for them to see because they don't see inside the red. And then when they walk through the chamber containing the powder, the powder sticks to them. And then as they walk throughout the hive, they leave the powder behind. So it's basically being inoculated. And the really nice thing about this is this fits right on the front of a hive, and you don't have to open up the hive at all to add powders. We have little inserts that you can change now. That all came about from feedback from other beekeepers, but it's just a really simple way to get things into the hive. And so that is kind of what got us started on this entire journey, was this collaboration with Dr. Kevin at the University of Guelph. Now, the advantages of bee vectoring are maybe not obvious right away, or maybe super obvious depending on you know, your love for going into hives. But the nice thing about bee vectoring is that you don't have to disrupt the hive. Every time you go into the hive, you affect queen laying. You can possibly squish bees. You can possibly kill your queen. If you're in small hive beetle area, you might be breaking the propolis gels open where the small hive beetles are escaping. And so bee vectoring allows you to quickly just add a powder in order to go in. And we always make sure we make it very clear. We're not telling people never to go into their hives. Just you don't have to go in all the time to do the things. So this is advantageous for that. It allows for very evenly distribution around the hive, and we've tested that over a couple of years, and we know that we do have great distribution throughout the hive. And if you didn't want to buy one for every single one of your beehives, with bee vectoring, you can actually move it around your apiary. Because it has a lot of different uses, most people buy as many of our ProtectaBEEs as they have beehives, but not necessarily necessary. And it's fast. So you basically open a drawer, you dump in your powder, you close, done. And, you know, we don't ever recommend people don't put on their suits with beekeeping, but you don't really have to smoke or get yourself ultra protected to do this, unless you're somewhere that you have bees that aren't very nice. Up in Canada, we have like really nice bees. So we, you know, I don't work with a lot of protection most of the time anyways. So just want to show you, as we did this for a couple of years, we went through lots of iterations of our ProtectaBEE. And you can see on the left there, we use a carrier powder usually for bee vectoring, and then we mix different things into it. So we tested antibiotics, Oxytet, that's used for American foul brood. We tested some different fungi. and we also tested Super-DFM. So these were all parts of our tests that we did to see if our product worked. And it did. It was fantastic. We couldn't have asked for a better outcome. The bees don't remove the powders. And if you compare it to conventional applications of the antibiotic Oxytet or Oxysol that's usually just spread over the brood frames like Slava was showing in his presentation, we actually see the identical disbursement of the antibiotic throughout the hive. So really could not ask for any better results. And the nice thing is that, you know, for beekeepers who want to add things quickly without disrupting the hive, this is just a really nice and easy way to do that. So today I decided to have a little experiment. So we've been testing Super-DFM, but I've never done like a time lapse to see how quick it would work. So we have all these volunteers that work best for bees, and so I had a couple come and grab some Super-DFM this morning. I said, do a time lapse for me. I want to see how long it takes for them to clean out the drawers with the Super-DFM. Now, the Super-DFM, it is a little bit different. It doesn't necessarily need to be tracked through the hive. The bees do eat it. It's got some sugar, sucrose, I think, in it. Like it's sweet. They want to eat it. But we also, if you look at some of the close-ups, we definitely have some bee vectoring happening with this as well. And I just want to show you the difference here. Our top hive here, this is Melanie Nobre. She's one of our participants today, and thanks Melanie so much for doing this. I messaged her this morning like, hey, can you do this with your hive? So she's got a hive that is fairly small. Unfortunately, they swarmed a little bit earlier in the season, and after she had done a split, they then decided to swarm anyway. So she doesn't have quite as many bees in that hive. And so we went ahead and tested her hive. And then on the bottom here, we have Michelle. She's another one of our volunteers, and she's got a hive that is happening. So we're really late. Our beekeeping season here, our bees really don't start until like mid-May. And so you can see how big her hive already is. They're bringing in the flow. We've got lots of supers on there. And so you can see there's a difference with how quickly the drawers are emptied out. So with Melanie's hives on the hot top here, 0 hours, 1 hour, 2 hours, 3 hours, 4 hours, by 5 hours, it's almost completely empty. And I don't know if she went back and did a 6 hour. That was, I think she started that around noon our time. So they're still going. Whereas Michelle, I was like, oh, it'll probably take you like 3 or 4 hours at least. She wrote me back after an hour, sent her photo, and she says, this is 1 hour. And there's just like barely anything left in there. And then she sent the 2 hour just for the sake of saying, yeah, and they really did finish it off. So this is a really good method for getting the Super-DFM honeybee to the bees. And it's actually placed on, if you look at our ProtectaBEE here on the far right, you'll see it's only actually placed on one side, and that's the indoor. So we have basically an indoor and an outdoor. We don't want it in the outdoor because we don't need them delivering it out to flowers or anything like that. We want them to bring it into the hive. So all of these, you'll notice the pictures, the cones are going in, so they were put in the indoor. But anyways, it was a little fun to do that experiment today just for our presentation. And thanks so much to my volunteers for helping out there. So I don't know if you guys have this, if this hopefully is playing for you. Sometimes the internet can be a little bit weird. But what we ended up doing, we originally started this off as bee vectoring, but our beekeepers that we tested with, they asked us to make inserts that would do different things for the hive throughout an entire season. And so we did that. We've got entrance reducers, we've got solid ones if you need to close up your hive for a short period. And you can turn the cones in to limit outbound traffic. But some of the cool things that we found out that were completely unexpected also with our ProtectaBEE is that robbing issues went away. So the wasps, hornets, yellow jackets, they actually don't see in the red spectrum. And this also removes the smell of the food source, the nectar and honey, a little bit further away as well. And so we find that robbing issues are not a big deal. We don't have robbing issues really with this system, which is fantastic. We also have Michelle, that big hive that we saw there. She has all sorts of visitors to her hive each night. She's got raccoons and possums and mice and skunks. And before we put the ProtectaBEE on, the skunk would be hitting the hive and the bees would be coming out and it would be eating it. And we put the cones on about 2 weeks ago, and the skunk is completely ignoring that hive now. So it can deal with those type of predators too. So what we expected to just be bee vectoring, and that was how our experiment started. We've ended up with this product that does a whole lot of things for a beehive. And if you're in the midst of a big flow, robbing's not an issue. You can actually take the inserts off, and then you have a super large landing board protected from overhead predators. And if you have like wind issues, it also protects them from wind as well. So just a lot of good things happening with the ProtectaBEE. And now, oh gosh, we can put our probiotics in there too. So even better. So just this last week, we've connected with Strong Microbials, and we have added their products to our website. So if you want to come and order some of that, you can do it off of our website. And I've even added on a Super-DFM ProtectaBEE combo. So if you want to get them together, you can find them there at our website at bestforbees.com. And I just want to say a huge thank you to the team at Strong Microbials. They're going to be talking with us a little bit. So if you can go ahead and put your questions in the chat box, and I've got some questions for you guys too. So we'll hopefully get some good questions happening here. And also, if you guys have other things that weren't in your presentation and you just want to share them with us, that would also be great. So. All right, so let's see what we've got for questions. If you have not put your questions in here in the chat, you can put them in. And I see something here about any differences in small hive beetle. So that's from one of the three Renees. Which one is that from? Is that from?
That's from me. Hi, I'm Renee.
Are you asking about my product or with your product, the difference?
Your product. They also don't like the color red. I don't know if solid red. And I know you probably don't have much hive beetle up there.
No, so we actually, there's small hive beetle about an hour away from my research apiary. An hour away, but not at my research apiary yet. And that sounds like a bad thing, but because we wanted to do the research, we can't. So we're coordinating this year with beekeepers in small hive beetle areas to see if it works for it. And we've also incorporated a brand new monitor in it as well that has an opening just small enough for a small hive beetle to go into, and you can bait it and then just pull it out at the front and see if you have small hive beetles in there. So we're hoping it'll eventually be a trap, but right now we're just calling it a monitor because we haven't been able to test that it would actually work, for being a large scale trap.
Very interesting. Thank you.
Yeah. So, all right. So I have some questions with you regarding, there's two different products and I put them both on my site. We have Super-DFM Honeybee. And we have Super-DFM plus P108, I think is the number.
801.
And
801.
Is it 801? 801. Okay, there's a little dyslexia there. So 801. Can you guys tell us about what the difference is between those two? And I know Renee has said that, and the real Renee, not the other two impersonating Renee. I know you were saying something about that it's not approved in Canada yet, but it's possibly coming to Canada. And I'm sure we do have a lot of people here actually that are from Canada, so they would want to know a little bit more about that as well. So if you guys could tell us a little bit about the difference between the two, and then where they're being sold and what's coming up. And there is John. All right, welcome.
Hello, John.
Hello.
All right.
Whoever wants to take this question, or I can, by all means, please.
Yes, so we have actually 3 versions of DFM we have right now. One is the Super-DFM powder, which we saw with your product. Another one is the P801, and that is actually the addition of 1 extra bacteria, Pediococcus acidophilus. And that is what is really going to help with pesticide ingestion and more defense against more of the, I would say the sorry, breaking up here.
Essentially, it's pesticide protection. We are able to protect bees from exposure to fungicides and neonics. It is a very specific product for a very particular application. We originally designed this product for commercial pollinators. However, number of hobbyists who live in areas that are surrounded by modern agriculture, where heavy sprays do occur, have asked us to create a smaller size, and we did. So it is definitely available. It is currently not available in Canada for the moment. We're in the process of finalizing our registration there. So probably within a month it will be available. Absolutely.
Okay, and sorry, Renee.
Super-DFM through Strong Microbials is the only probiotic registered for legal use in Canada.
Correct.
So everything is completely up and up, and absolutely fine with your government to work with.
All right, and we find we have about 75% of our customers are from the US and 25% are from Canada. Given that Canada is about 10% the size of the US, though, that's a pretty good representation of Canadians as our customer base. We have a question here from Rebecca. She says, food microscientist here. I'm curious if the microbes have an impact on human gut if ingested. Is it okay to be caught in the honey?
Of course. Whoever wants to answer it, or I will.
It shouldn't cause much of a problem. I mean, most of those microbes might possibly be beneficial to you, although I doubt much if all would get into the honey. I'm not sure about using the ProtectaBEE system. It'd probably be, I don't know, probably 0.001%, I imagine, just by happenstance. It'd be mostly consumed going up through the brood chamber and cleaned. There's always a slight possibility, though. And then during using our methods, the regular method of just sprinkling it on the brood frames, it shouldn't get in the honey at all. In the US, it's you're allowed to use it during honey flow.
We have tested this on our hives on top of our warehouse, and we produce honey off of there as well. And we have seen no ill effects on the honey that we consume ourselves. We're still alive.
Yes, a lot of our really big customers also test their honey because they are honey producers and they use our products, and not a single time have we ended up in honey. So all is well. Now, honey does have natural microbes in it, but it's not ours that end up in there. So whenever bees collect pollen and nectar, they do bring in microbes.
Now, a lot of microbes are already, like, you would find them in yogurt and things that people are consuming, right? These are not necessarily things that are unforeign to us in the foods, and it is part of fermentation, right? Is this right, that we would commonly come across this in the foods that we're eating?
Of course, yes. And everything that we use is on a USDA generally accepted as safe list. It's a GRAS list. This list, or these species, are maintained by AAFCO. It's an American Association of Feed Control Officials. So everything that we work with, everything that we use in our products is safe, non-toxic, non-modified, came from nature, and is, only beneficial. I really do not want to work with anything that will hurt myself or anybody that works with me.
Yes, this is not a medicinal product in any way. the bees will fight off pathogens and diseases by way of just being stronger from the inside out, but there are no medicinal claims, there are no chemicals, and everything in here, besides the sucrose, is already found in a honeybee's gut.
Now, I have a question. I know that when we look at the microbes we find in the honeybee gut, we actually find a lot of different microbes, not just a few, right? There's a lot. Have you guys looked at adding other microbes to future honeybee things? Are you guys testing it? Like what's the future look like? And are all the microbes that are currently, I'll leave that as a second question. Actually, I'll leave the first question and let you guys go with that one. And then the second question.
Absolutely. Thank you for this question, Eric. Absolutely. Yes, we are working on researching and potentially adding more microbes to our probiotic mix. Definitely. There are many microbes in the world, in the gut of the honeybee. We chose to work with the ones that are safe and most beneficial for production, longevity, and immunity, specifically those. There are multiple microbes, for example, in the gut that are either benign or opportunistic. And even though they're not pathogens, but they can turn into pathogens if an opportunity exists, meaning that, for example, if they were treated with antibiotics and the majority of the lactic acid bacteria, for example, were wiped out, then this opportunistic bacteria will see the opportunity to eat food and grow and colonize, and that can actually potentially hurt bees. So we don't want to add that into our mix just because it already exists in bees.
All right. Oh, that's great. So Slava, Melanie, who you saw her smaller hive there, has a question for you. And she says, you mentioned that the bacteria doesn't transfer to the honey, but what about the sugar in the product? There are very strict requirements on the standards for honey, and foreign sugars are a big no-no.
Mm-hmm. Absolutely. So the amount of sugar that sucrose that we're delivering is unbelievably small. We're delivering grams versus a large amount of money that honey exists. Also, whenever honeybees eat, they have, once again, they have a honey sac or honey stomach and the actual digestive system. So whenever honeybees partake of our products, it ends up in their digestive system as opposed to their honey sac.
All right, and that's good to know, because it's not something they're actually ingesting it as opposed to storing it.
Correct.
And why you guys are not finding it in the honey? It's not something that they're putting in there.
But that's a great question. Thank you for asking that. I appreciate it.
All right. Okay, this is a question for us at Best for Bees. So Nancy says, I understood you mentioned the ProtectaBEE does not need to be used in every hive in the apiary. How many should be placed on hives? 1 every 2 to 3 hives. And please, Renee.
That was my fault. I was trying to rename Slava and I hit the wrong button.
He kicked Renee out. No. All right. So Nancy, just to answer your question. So when we originally made the ProtectaBEE, our idea was that it would be for bee vectoring, and usually, as you see, the drawers are usually emptied out within 6 hours. So you could definitely just have a few and move them around your apiary. Now, what has happened is that we've added all of these inserts, which actually add a lot of other features to the ProtectaBEE. And so we find our beekeepers, our commercial beekeepers, their plan is actually to have a single ProtectaBEE on every single hive, which means they don't have to move things around, and they can quickly, change their entrance reducers. They can move the cones on and off, and they also have that protection from those other predators, the skunks that we mentioned. We actually have seen opossums actually trying to get into the hive. So and mice, the cones actually keep mice out as well, which are a big issue, especially in wintertime for bees. So you could definitely move it around, and the number that you would have would be how often can you get out to your hives to move it. You know, if it's 6 hours, you could maybe do a couple. The important thing though is remembering you've got to follow biosecurity measures, because if you do have one sick hive, you don't want to transfer it to another, just like you're cleaning your hive tool off in between hives as well. And so we recommend using alcohol and the ProtectaBEE if you're moving it from one hive to another. All right. Okay, so we move that to answer. Okay, I have a question. I don't see any here yet. So if you guys do have questions, there's a Q&A section or chat. You can put them in either one. So my second part of the question that I was going to ask is, do you guys have any probiotics that you started using and like one of the bacteria, and then you're just like, you know what, actually that's not having like amazing effects. Like it's actually not something we need to have in there, and that you've thought about simplifying or taking it out or replacing it. Has that happened along your journey, or is it something you see possibly happening in the future? And do we not have Renee? She's like gone forever. John, what'd you do?
We're working on it.
That's okay, John. I made your host, so please take care of this. I'm grateful. But Erica, to answer your question, yes, we did. When we first started developing our products for honeybees, we tried a number of different yeasts. So our microbes contain, our Super-DFM contains lactic acid bacteria, it contains spore-forming bacilli, contains yeast, and it contains enzyme. So we use yeast not only for just as a nutritional supplement, but as also as an oxygen scavenger, just to create an even more favorable environment in the gut for the lactic acid bacteria. However, certain yeasts were not very palatable, even for us. What I mean by palatable is just the particular smells they were generating, or just it was not palatable for bees. Bees did not want to eat that; they didn't like it. And we had multiple different yeasts that we tried, and we chose the one that not only that does the job of consuming oxygen, scavenging oxygen, but to also make it as palatable as possible. So that was one of them, absolutely. As for microbes, as for like lactic acid bacteria or bacilli, we haven't had to eliminate any yet, just because we did like really careful research about what to put in before we do it. Yeast, we had a variety to choose.
Okay. All right, let's see if we have any other questions. I do have some more questions for you if there's no other questions. All right. Okay, so my question for you is you have a recommendation. I've actually seen a couple of different recommendations depending on whose website I've been to or looked at. Some are recommending every 2 weeks to be adding in the powders. You guys had every 30 days. And as a microbiologist, so one of the things that you think about is if this has colonized the gut of these bacteria or of these bees, and then they're walking around, they poop, whatever, they should still have them there. Why is it not persisting? Why is it not staying around that you have to do it multiple times? Is it because of the bacteria themselves, or is it because of the exposures in the environment that the yeast sorry, that the bees are constantly being exposed to? Like, which part of it is that?
Is the second part, really. It's everything in our environment is dynamic. A lot of temperature changes, ultraviolets, pests, pesticides, treatments that we use, different things that bees bring back themselves, food, all of it, causes the gut to be dynamic. It's, for example, when food comes in and gets digested, part of that mucosa gets sloughed off and travels downstream and gets treated during the cleansing flight. New food can come in that, for example, will have slight antimicrobial properties to it. Cinnamon, for example, can have strong antimicrobial properties. I have seen beekeepers use cinnamon. seasonality, lots of different things. And it's not like, you know, they completely go away after a month. Absolutely not. No, there's a slight reduction. And what we see is a reduction. We see a reduction about, you know, 10 to 15% within the first month. So it's not like everything is gone. But the reason for why it's gone is because part of it gets used up. Microbes do divide and they do retain in the hive. However, once again, summer bees don't live as long. So yes, the microbes get spread around in the hive through trophallaxis, you know, oral trophallaxis to clean up the hive. But most important one is a fecal oral trophallaxis. Majority of the honeybees microbiota reside in the hindgut. And these are social insects, so whenever they go on a cleansing flight and come back, others help clean up, essentially. And that's one of the main ways how microbes get transferred from bee to bee. Eventually, it's a law of diminishing return. You use them up. You have to resupplement again, and resupplementing either using our products or bees go out and forage and they bring in new ones.
All right.
Excellent question. Thank you.
Yeah. All right. I'm going to ask you a question not about bees.
Hmm?
So, because a lot of our people are, one of the things I found out anyways, when I started, we started doing our crowdfunding campaign for the ProtectaBEE, is we had to look at audiences. Who are the people that are interested in your product? And our biggest audience by far was our homesteaders. So we think that the majority of people, not the majority, but a lot of the people who have bees also have backyard chickens and other small livestock. What's the difference? Because I know you guys have Super-DFM for some of the other animals. What's the difference between those, and how often are you having to give? Are you having to give them as frequently or less? I'm just kind of curious how that works in other animals.
Of course, differences in microbes depends on their diet. For example, if we're probiotics for dairy or for any ruminant animal, we'll have another microbe called Propionibacterium. That is a necessary microbe that is typically found in ruminant animals. Bees don't have it. They don't have a rumen, so they're not going to have that bacteria to them. Chickens, for example, there also depends on their diet. They have a particular microbe, you know, that's specific to chickens called Lactobacillus reuteri, for example, and we're tailoring the microbes to be species specific. A lot of the microbial species are conserved on this planet. You know, for example, Lactobacillus acidophilus is found pretty much everywhere. In us, in bees, in fish, and mammals. So like you name it, it's quite conserved. But some are quite specific depending on the diet. Frequency of application for production animals, for example, we recommend applying it almost daily, just because they're much bigger and they are able to encounter more challenges or less hygienic. For example, chickens are coprophagic; they peck at their own fecal matter. So okay, hogs, for example, also are in an environment that can be less sanitary than desired. Dairy cows as well. I believe bees are very clean, and and based on our research, once a month is more than once a month of our application is more than necessary. But I really like your application because it's a continuous inoculation, and that's perfect.
Makes it easy. All right, so we've gone over the 8 o'clock time that I mentioned to you guys we were going to be wrapping up. I was having so much fun. I wasn't even paying attention to the time, which is pretty much my life.
It's all good.
So I want to just say thank you so much. And I did hear you on Frederick Dunn, The Way to Be, like you were like the same week as me chatting, and it was just really interesting, this crossover. And you know, we're working with microbes as well, but we're, you know, focused more on fungi, but we may be coming over to bacteria as well. Anything that we can vector with our system is a possibility, and we're collaborating with lots of scientists across the world. And we're just happy we can bring your product to our site, and hopefully a new way for you guys to also, have your product applied. It makes it nice and easy for people. And yeah, so I just want to thank you. We didn't introduce John. John's been there trying to get Renee in for the longest time. There she is.
There we go.
Hi, Renee. Sorry to kick you out.
I'm never
That
allowed to be
works.
host again.
There she is. She's joining us for the end. So a huge thanks to Renee. Renee's like a tried and true farmer who's been working with Slava and Vera for several years. I know John's involved in the sales portion of it, and Slava's CEO and doing all of the research. And I'm just so thankful you guys were able to join us today. I hope our people that have joined us got something from this, and we recorded it. So people for, as long as we have the internet and YouTube, can continue watching this and learning all about probiotics and how they can improve the health of bees. So thank you guys. Thank you so much. And thank you everyone for joining us today. I really appreciate it. And stay tuned. We'll be having more chats with different scientists and different products as time goes by. So thanks so much for joining us.
Thank you, Erica. Yes, thank you, everyone. All the best.
Bye. And thanks, Renee, for joining on the phone.
Bye-bye.
All right. Bye.
All the best.
Thank you.
Have a good night, everyone.
Late and kicked you out.
Have a good night.
Do I have to do something here as host?
No, this is super weird. I'm gonna go drink now.
Sorry about that.